If you alter the nucleic acids, you alter the proteins those nucleic acids code for. Welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, Science and Technology. First of all, please like this video, please share it, and please subscribe to my channel. Well, I wanted to come back and make hopefully a quick video on the concept of mutations at the molecular level and also at the subsequent protein level. I think it's a timely discussion as it directly relates to the world event that we are going through right now, but it also, in terms of the biological sciences and human health, it has a it applies there as well. So I thought this would be a fun video to make and a fun discussion to have here on my channel. So with that, let's talk about mutations. Now, there are several contexts for mutations, and as I'm talking about this, I'm thinking about pop culture, and something you may all be thinking about is Marvel's X-Men, where you have a population of mutants constantly at war with the non-mutant population. But when you think about mutants and mutations, in general, what you want to think about is something different or something altered. Some mutations you can see openly. If I recall correctly, well, I'm not going to speculate on that. I'm just going to say that some mutations you can see physically on a person or an organism in terms of phenotype, and some you can't see. In some instances, there's a disease state that's caused by a mutation, such as in certain populations, cystic fibrosis, and in some instances, there are non-harmful mutations, and mutations occur not only in humans but also in other animal species, and even down to the bacterial and viral levels or spheres, whatever you want to call it. But there are several contexts and aspects for this, and in the biological sciences, we're normally discussing these in terms of nucleic acids and the proteins that they give rise to. So with that, I'm going to have a short discussion on that. Now, I've stated throughout my content on this channel that proteins are the workhorses of the cell, of our cells, and those could be enzymes, those could be receptors, they could be channels, they could be proteases, they could be cytoskeletal proteins, transcription factors, any number of things. But those are what actually they actually do the work in our cells, and we know that those proteins are coded for in terms of their amino acid sequences. Those are coded for by our nucleic acids. The DNA gives rise to RNA, and the RNA goes into the cytosol. So I created a video on that, and you can go back earlier in my library and take a look at that, but I just wanted to refresh us and say that our proteins are the workhorses of our cells, and if something abnormal happens to our proteins, whether it's chemically induced or if it's as it's being transcribed, if it's transcribed and translated incorrectly, that can lead to a mutation and also a disease state of some sort. Okay, so it ultimately goes back to our nucleic acids, depending upon the organism or the microbe or the contagion, whichever it is. The proteins, the protein sequences come from the nucleic acids, whether that's DNA or RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. They have similar properties, but they have different properties and different structures. That's in my earlier video. I'm not going to get into that. But what I wanted to say was that, or should I say what I wanted to impress upon those viewing this video, and this is, folks in the biological sciences know this cold turkey, as one of my professors at Johnson C. Smith University used to say, but the sequences for the proteins come from our nucleic acids, and if there is some alteration to these, to our nucleic acids, that can lead to damage of the proteins themselves, or it can cause the proteins to be created in a damaged and altered way, which can lead to a disease state. So to illustrate what I just talked about, I'm going to show this slide here, and it was asked that attribution be given to the creator of this slide, and so I want to give attribution to Mikey641. This slide involves what we in the biological sciences call point mutations. And a point mutation is simply when you alter one small piece of the DNA sequence or a DNA sequence to change the identity or to change the amino acid in a specific protein sequence. And so if you look at this slide here, you see the first set says no mutation. The DNA base pairs, they're usually in threes, sets of three. So you have thymine, and cytosine. So the corresponding mRNA transcript is adenine, guanine, AAG, and the amino acid that codes for is the lysine. So that's no mutation. So you want to compare everything else on the slide to that. So a silent mutation would be substituting a third thymine for the cytosine. So you're going to have thymine, so TTT. And the corresponding mRNA transcript would be adenine, or 3 A's. And once again, the corresponding or the resulting amino acid is a lysine. You can also have a nonsense mutation where you have, in this instance, an adenine, thymine, and a cytosine. Your corresponding, or should I say your resulting, I'm interchanging those, mRNA transcript is a uracil adenine guanine, which is leading to a stop in the generation of that protein. You can also have a missense mutation. You can have a conservative missense mutation where you have a thymine, cytosine, or a TCC. The corresponding mRNA transcript is adenine, guanine, and guanine, and your resulting amino acid is arginine. Now, amino acids, there are groups of them. You have polar amino acids and nonpolar amino acids, and you have acidic amino acids, and you have more basic amino acids. So you should notice that the arginine and the lysine, I'm just going to keep it simple, they have similar chemical properties, and I'll stop there. You can have a non-conservative missense mutation. So you have a thymine, guanine, and cytosine, and your resulting mRNA transcript is adenine, cytosine, and guanine, and that is giving rise to a threonine amino acid. And if you look at the structures, the lysine structurally is similar to the arginine, but the threonine in terms of structure chemically is very, very different than either of those two. So that would likely give rise, that would likely have a more deleterious or significant impact on the protein that's being translated in the cytosol. So those are point mutations. In the biological sciences, that actually is a tool that scientists use to characterize protein structure and function, and in some instances, disease states. So scientists use point mutations to understand, for example, how the active site of an enzyme works, or how a channel or a receptor works. You sequence that protein out, you identify the regions of that protein, you see where the polar amino acids are, where the nonpolar amino acids are, where the acidic ones are, where the basic ones are. You think about where the active site is, and you might want to figure out what the key amino acids in that active site are, or the key amino acids in that receptor channel are. And scientists oftentimes, if they see, for example, there's an acidic amino acid in there, they can substitute in a more polar, a more nonpolar amino acid in there, and then look at the effect of that substitution on the activity of that enzyme, or the ability of that channel or that receptor to perform its function. So in some instances in the lab, being able to substitute out key amino acids can give valuable information about how a particular enzyme or receptor or channel or transcription factor, works. So this is a scientific tool in addition to being a phenomenon in nature. So how do mutations occur? Well, I just described that in the laboratory research setting, mutations can be used as investigative tools, and there are, too many papers to name where researchers have used point mutations to characterize how a protein of interest inside a cell or a tissue works. But mutations also occur in nature. They can occur randomly, though I will say that our cells, our eukaryotic cells, actually have enzymes and mechanisms to ensure that DNA structures stay intact. So there are corrective and protective mechanisms within our nuclei to make sure that, or to ensure that our DNA structures stay intact, and likewise that ensures that the proteins they create will likely be functional and healthy. But they occur in nature depending upon the species and depending upon the organism. In some instances, as a pharmacologist / toxicologist, in some instances, chemicals can induce damage to DNA, which can lead to mutations in the proteins that they create, which can lead to disease states. I'm sorry, which can lead to disease states like cancer. So that's one context. Radiation from the sun. We know that our atmosphere filters out harmful rays or aspects of the electromagnetic spectrum, which can cause DNA damage. And so gamma rays, for example, or ultraviolet radiation, if I recall correctly, can damage DNA in our cells or for any organism, and that damage can lead to, once again, a disease state like cancer. And also in certain organisms, as I said, depending upon the organism and where it sits, some organisms mutate more readily than others. Earlier in this video, I mentioned the disease state cystic fibrosis, and if I recall correctly, that exists predominantly in Jewish populations. I recall doing some research on that as an undergraduate. That is considered an autosomal recessive disorder. And if I recall correctly, it occurs in the respiratory tract. There's a transporter. The gene is referred to as the cystic fibrosis transmembrane conductance regulator, and the most common mutation is the deletion of phenylalanine 508, and that results in an altered protein and the ultimate disease state that individuals born with this condition experience. I believe, as it's in the respiratory tract, I believe that it's there's trouble breathing, and these people cough, and there are abnormalities there. So I'm going to conclude with a discussion of our current world event. But first, I'm going to point you back to my video on the Western blot analysis, the one with the more complete and in-depth discussion. Towards the end of that video, I discussed how antibodies work and how antibodies are used in the Western blot analysis. And so what happens is, what antibodies do and how they work is that the antibodies recognize specific epitopes on the protein of interest. So that's how the antibody identifies and attaches itself to the protein of interest. But what happens if there's a mutation of some sort and that epitope changes? Well, then that antibody cannot recognize that new protein. So you might have a contagion floating around in the population around the world that is randomly mutating. And it could be that you've created a medication such as a vaccine against that contagion. Well, if that contagion is mutating regularly and the epitopes on those proteins that those antibodies were recognizing have changed significantly, then your treatment, your antibody, will likely not recognize that that contagion anymore, and so you have to come up with a new medication. I'll just say that. I think you guys can read between the lines of what I am saying here, but this is a significant concern in the medical community, and whenever there is a contagion of interest, a key question is it mutating and how often is it mutating, and how long will these medications that we've created, how long will these medications for the population be effective? And I did some reading before I shot this video, and it turns out that depending upon the contagion, some contagions mutate rapidly, while others mutate slowly. And there are multiple ways in which contagions can mutate. So again, there are lots and lots of contexts for mutations, and I wanted to just give an overview of those from a research perspective and a nature perspective, but I wanted to close that out, or I wanted to close this discussion out with how this relates to our current world event. All right, everyone. Well, that's my discussion on mutations in terms, or should I say in the context of nucleic acids and the proteins that they ultimately give rise to. It's possible, I left something out. If so, there may be a follow-up video to this. But please let me know what you think in the comment section below, and also if you have any questions in the comment section below. If you want to leave something in the tip jar, that information is below this video as well. And I'm going to leave, if I can find some, I'll leave some pertinent articles there as well relating to what I've discussed here in this video. If you haven't, please subscribe to this channel. I am approaching the 1,000 subscriber mark, and I need a few more, and every subscription helps. So with that, I'm going to stop. Please like, share, and subscribe. As always, remember that your attitude determines your altitude. Take care, and I will talk to you the next time. Bye-bye. Oh, by the way, guys, I forgot to mention this in the first part of this video. When a contagion, and I'm being very careful about using that word, when a contagion undergoes a certain amount of mutations, it can become designated as a variant. So in the immunology and in the virology world, that distinction gets made, and that's a key vocabulary word. So when you see that, it's usually referring to a contagion that has undergone a certain number of mutations.